Human reproduction begins with a long journey. After fertilization in the ampulla of the uterine tube, the embryo, now a hollow sphere of cells called a blastocyst, travels into the main uterine cavity. But arriving in the uterus is only the first step. To survive, this microscopic sphere must forcefully embed itself into the maternal tissue. This is the exact definition of implantation. The clinical text notes a strict two-part sequence: initial attachment to the endometrium, followed by deep embedding. This critical window is brief, taking place between the 6th and 12th days after fertilization. Over these 6 days, we will track the biological sequence required to transform a free-floating cluster of cells into an embryo permanently anchored within the uterine wall. To map the maternal-fetal connection, we have to examine the precise tissue alterations and cellular maneuvers that make this survival possible. Before any physical contact occurs, the maternal tissue must prepare. This begins with the decidual reaction. The endometrial stroma undergoes specific structural and biochemical changes, swelling with nutrients in direct anticipation of the blastocyst's arrival. Meanwhile, the embryo faces its own obstacle. It is still trapped inside a protective outer shell called the zona pellucida, and it must undergo hatching. On the 5th day, the blastocyst begins producing an enzyme called trypsin to attack this barrier from the inside. Trypsin causes the zona pellucida to thin out, leading to its complete structural failure by day 6. As the protective shell completely dissolves, the internal mass of trophoblast cells rapidly bursts free into the open uterine cavity. Shutting this barrier is a mandatory trigger. Without the zona pellucida in the way, the embryo and the mother are cleared for direct cell-to-cell interaction. Now exposed on day 6, the embryo achieves its first physical milestone: adhesion. Specific cells responsible for establishing this contact are known as the polar trophoblast. These polar cells are grouped at one end of the blastocyst, situated directly over the inner cell mass, or embryoblast. This localized cluster makes physical contact, sticking firmly to the columnar uterine epithelium lining the mother's tissue. This initial adhesion serves as the formal trigger for penetration. The polar trophoblast cells immediately begin forcing a passage down through the uterine epithelium, pulling the entire blastocyst inside. The moment adhesion occurs, the biological priority shifts from drifting entirely to invasive mechanical burrowing. To burrow deeper into the uterine mucosa, the blastocyst must erode the solid tissue blocking its path. It accomplishes this using targeted biochemical weapons, proteolytic enzymes. This erosion requires a localized chemical breakdown. The destructive enzymes are secreted collaboratively by both the embryonic polar trophoblast and the maternal uterine epithelium itself. By day 8, the embryo is partially embedded. To continue advancing, the trophoblast must undergo a major structural differentiation, splitting into 2 highly specialized layers. This diagram shows the formation of the first differentiated layer, the cytotrophoblast, or Langhans layer. This inner ring consists of distinct mononucleated cells packed with active mitotic figures. Their sole function is rapid division, continuously generating new cellular material. The cells generated inside are pushed outward, fusing to form the leading edge, the syncytiotrophoblast, or plasmodial layer. This outer layer is entirely different. It is an amorphous multinucleated mass that completely lacks mitotic figures. It does not divide. It rapidly expands to dissolve the surrounding maternal stroma. This elegant 2-layer system solves the mechanics of deep invasion. The inner layer provides constant structural proliferation, while the outer layer handles the aggressive, path-clearing destruction. As the embryo pulls itself completely inside the uterine wall by the 9th day, it leaves behind an open penetration wound in the epithelium. This structural defect must be sealed. It is quickly filled by a coagulum consisting of tissue fluid and cellular debris. This biological patch forms what is known as a fiber plug, completely closing off the embryo from the main uterine cavity. We finally reach the 12th day, marking the absolute completion of the embedding sequence. The outer syncytiotrophoblast has now burrowed deep enough into the stroma to reach the maternal blood vessels, specifically the maternal sinusoids. The amorphous outer cells force their way directly through the vessel walls, tapping directly into the mother's circulatory system. Breaching these vessels officially initiates the vital nutritive relationship between the mother and the fetus, marking a successful implantation cascade. Up until now, we have tracked implantation purely at the cellular level. Now we zoom out to map these events across the entire uterine anatomy. The biological mechanics we just covered classify normal human implantation as interstitial. Interstitial means the blastocyst is completely subsumed within the endometrium of the uterine wall. This is distinct from central or eccentric implantations in other species, where the embryo remains exposed in a cavity or a crypt. Statistically, this embedding follows strict spatial patterns. Normal implantation overwhelmingly occurs in the upper part of the uterine body. Within that region, the blastocyst slightly favors the posterior wall, occurring there 55% of the time compared to 45% on the anterior wall. However, this targeting system carries severe risks when deviations occur. The clinical notes categorize 3 primary abnormal sites of implantation. If the blastocyst embeds in the lower uterine segment, the extrauterine tubes, or escapes out into the abdominal cavity, the result is an ectopic pregnancy, a condition with massive obstetric complications. Implantation requires highly orchestrated, localized tissue deconstruction. Securing the precise anatomical location and successfully executing the cellular invasion is the critical test required to secure the embryo's survival.